today we're going to do a demonstration on how to do a packed cell volume reading and a total protein reading on whole fresh blood in an EDTA tube. So we'll go over the supplies that we have here. So the first thing we have is our little centrifuge. We're going to use that to spin down the microhematocrit tubes. We have our fresh blood sample. We have our little stat spin tray that we're going to use to put in our microhematocrit tubes. We have our clay sealant. I have some clean Kim wipes to clean up afterwards. I have some distilled water also to help clean up. I have my refractometer. And I have two different little reading charts for reading our microhematocrit tubes. Either works fine. I prefer this one, so I'm going to use this for a demonstration. Okay, so we'll get started. So in a real case scenario where you want to actually get a true diagnostic reading, you want to first get a fresh blood sample properly collected, usually from like a jugular vein. And you want to process it immediately. So assuming that we just obtained this sample, the next thing we're going to do is we're going to take our fresh collected blood, we're going to invert it 4 to 5 times, so that just means gently rocking back and forth 4 to 5 times. You never want to do this aggressively, or you never want to shake it. That can actually damage the red blood cells. So once you do that, we're going to go ahead and we're going to fill up our microhematocrit tubes. So because I have a little blood in the cap here, I'm just going to set my tube aside and I'm just going to do this here. So we're going to fill the tubes up to about that little blue mark, and that blue mark is kind of like our, just over 3/4, 2/3 of the way. We're just going to fill it all the way, and we're going to seal the end with our thumb. I'm going to switch sides, and then we're going to seal it with the clay. And you're going to want to put it in your microhematocrit slot here in your spinner, and you want to put it with the clay side out. So I'm putting it in slot number 1. Okay, we're going to fill the other 3 tubes the exact same way. So filling it, your tube should look right about like that. I'm not sure if you can see that, but I'll bring it up. The tube should look kind of like that. And when you place it in your spinner, you want to make sure that you're balancing it out. So I put it in slot 1 first. I'm going to go directly across, and I'm going to put it in a slot directly across from it, which is slot 7. And you want to do 4 samples at a time and obtain 4 readings. And just make sure that they all match up. Seal it, put it in our slot here. Okay, so we're going to put this one in slot 10. And we're going to put our final one across from the last one we just placed, and that's going to be in slot 4. Oops. Okay, the next thing we're going to do is we're going to put our cover on top. We're going to make sure it's nice and seated, nice and tight. So before you put it in the spinner, it should kind of look like this. It's all symmetrical. Each tube is across from another tube, so that way it all balances each other out. So the next thing we're going to do is we're going to put it in our little centrifuge here. I'm going to stick it in. And we're going to make sure it's on the correct setting. So this particular little centrifuge is made specifically for these kind of things. So it actually has a microhematocrit setting. So we're going to make sure it's on that one. Press the lid down gently, and you're going to press the go button, and it's going to do its thing. Okay, so now that our centrifuge is done spinning, the next thing we're going to do is we're going to open it and we're going to go ahead and we're going to take our tubes out. Looks like they're all nice and spun down. And the next thing we're going to do is we're going to read it. So like I said, you could use either chart, either is totally acceptable. I'm going to use my circular one, it's just a matter of preference. So we're going to read all 4 tubes. And the way we do this is you basically line up the bottom 0% line with the bottom of the red blood cells, so the red layer. And then you're going to line the very top line, the 100%, with the top of the plasma layer, just between the plasma and the air layer. So we're going to go ahead and I'm going to read this. Okay, so I'm getting about 52% for the first tube. And again, you want to read all of them. You want to make sure that they all match up. Just in case you made an error in the first one, you want to make sure that you're getting the correct results across the board. Okay. So again, you're going to line the top of the plasma with the 100% and then the bottom of the red blood cells with the 0%. And again, I'm getting about 52. And we're going to read the third tube. Getting about 52%. And I'm reading the last tube. And so just so I can explain what I'm doing here, I'm actually reading, basically, I just stated how to line them up, but the way you find your number is you read the top of the red blood cells, where that one lands when you line up properly. And across the board, I'm getting about 52, 53%. So I think we can safely say our packed cell volume is about 53%. So the next thing I'll do is I'll pause this video and we'll go ahead and we'll do our total protein reading as well. And you actually do this with our tubes that we have here.